Step 1. Open the kit and remove the diluent. There are 3 bottles of diluents in this kit. Step 2. Remove the test kit and place it flat on the dry desk. Step 3. Add specimen. Take serum as example. First, draw blood specimen. Draw vertically blood into the specimen well, serum or plasma, 10 microliters, whole blood, 20 microliters. Second, add diluent. Add vertically 2 drops of diluent into the specimen well. Step 4. Read the results. Observe and record the results within 10 minutes. Interpretation of results.